Oh.
So did you see like this?
What was that? Oh, there. That was the recording starting. Okay. So go ahead and get your notes out. We will be talking about lipids today. So the reading that covered lipids was pages 53 through 55. And I will reiterate, like, I appreciate y'all telling me, hey, I didn't do the reading, that type of thing. I really do appreciate it. Just know I'm not, like, grading you over your reading or anything. It's just you get out what you put in. So, you know, if there's something that's unclear or anything and you need to go back and do the reading, it is however you best learn the material. I just like the reading before, so that way those that do the reading can come in with discussion and things like that. Again, this is a college level class, so I'm not going to hold your hand and be like, okay, you know, I'm going to grade you over your reading, that type of thing. So anyways, it's just basically habit forming. And again, nobody's over procrastination because I listed my house this weekend. And so I literally I submitted my lesson plans yesterday that were due on Sunday. So like it happens. Okay. We are all human. Okay, let me see. Can y'all see that color okay? Or do I need something darker? Here, hold on. Let me get something a little bit darker. Where did that darkness go? I literally just had my pen. Oh, there's a black one that'll work. Okay. Okay. So, y'all listen up, please. So, what do you recall about lipids? What are they? They're fats and oils and waxes. So lipids are the second group of macromolecules. They're called not a true polymer. Why do you think that they would be called not a true polymer? It didn't really explicitly say this, but I just want to see.
I wrote down that they're not true polymers, but I don't know why.
So a polymer, by definition, is just simply a polymer built up of monomers. So basically, those building blocks in a general definition of a polymer would be the same. The building blocks in a lipid are different. So meaning that it's not all a monosaccharide combined together like a carbohydrate. It's got two distinct different types of monomers. Does that make sense?
Totally.
That didn't sound very convincing. So, like I said, with carbs, so like where carbs are, all of them look generally the same, blah, 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 and they're all built on top of each other. But those building blocks obviously aren't the exact same, but they all contain, carbons, hydrogens, oxygens, things like that. The lipids, they've got your glycerol backbone, and it's not a cyclic molecule. So here, hold on. I'm going to draw this bigger. But it's got a carbon, hydrogen, oxygen backbone that looks very similar to carbohydrates, if you recall. Except that's an H. Goodness gracious, get it together. This is one part that is glycerol. All that means is it's a 3-carbon sugar. So that it's literally got a carbohydrate backbone, and then it's got its fatty acid chains. I think I went the wrong. Yeah, I went the wrong way. Sorry. So I'm just drawing a short chain. I'm not being precise on carbons. Abbreviated, a lot of times you will see these tails look like just squiggles. You just need to understand that at each of those points is a carbon, and that every carbon has If it's combined with another carbon, then it's got 2 hydrogens. If it's, at the end of the chain, you're going to have 3 hydrogens, or, another carbon containing group, that sort of thing. But just know that abbreviated squiggle is a carbon at each point with hydrogens connecting. And we'll go ahead and tack that second one on there. So what it means by saying it's not a true polymer, and again, that's not lipids, so I don't want to confuse anybody. It's just showing you an example. But what it means by not a true polymer is that the building blocks are very, very different in composition. So one is an actual carbohydrate, glycerol, and then the second is a fatty acid chain, is all that means. What's so special about lipids in reference to water? They're hydrophobic. What does that mean?
They hate water.
They hate water. So that's super important because that is one of the main properties of a lipid that have to do with its function. And again, it's hydrophobic. Why? What makes it hydrophobic?
The non-polar carbon.
Exactly. So this fatty acid chain right here is nonpolar. So it just means neutral charge. It doesn't have any positive or negative end associated with it. And so it doesn't mix with the polar water molecules. So that is one of the main properties as why lipids act the way they do. And just going back to the whole unit of structure and function, because it's this structure, it has a specific function. The reaction that combines them, just like we talked about with carbohydrates, that's giving off water. And it combines, just tacks on to one of those oxygens. I'm not going to go into like the specific chemistry or anything like that. There's no double bonds moving or anything. It just tacks it on. And the bond itself is called an ester linkage. I don't care that you know that it's an ester linkage or anything along those lines. And that's not of super importance. Just know whenever you're building up a lipid that water's given off. Just like we talked about last time, whenever any polymer, so whether it's carbohydrate, lipid, protein, nucleic acid, anytime those are built up, water is always, always, always given off. It's always a dehydration reaction. Okay, so there are a couple different types of lipids. So there's fats, and then there is phospholipids, and then there's steroids. It covers briefly on steroids, and we will cover briefly on steroids. There's a little application to that we'll talk about later, but right now we'll just talk about why it's considered a lipid. So regular fats, there's a couple different kinds, saturated and unsaturated. What do you recall about saturated fats? What does it mean to be saturated? Oh, they're so quiet today. So not so much dissolved, but it means that basically all the bonds are met. So it's completely saturated with hydrogens. Oh my gosh, I can't speak. With hydrogens, completely saturated with hydrogens. That means that it's only single bonds. So a saturated fat, the fatty acid chain is going to look like this, where it's got all its hydrogens, and it's completely saturated. There are no double bonds. This is where it connects to the glycerol. I'm not going to draw it out, but anyways. So this is your fatty acid chain. This is saturated. That just means it has all of its available electrons taken up by hydrogens. Completely saturated in hydrogens. So what do you think unsaturated means?
At
It loses hydrogen. So what happens if it loses a hydrogen? What do those bonds have to do? And so single bonds, you get double bonds. So what I'm drawing right now is a specific type of unsaturated. We'll talk about it. And again, I'm only drawing one H because if you look at a single carbon, sorry, let me zoom in there. If you look at a single carbon, there's 1, 2, 3, 4 bonds. 1, 2, 3, 4 bonds. 1, 2, 3, 4 bonds. So each carbon needs to have 4 bonds. This one doesn't have as many hydrogens because it's taken up in double bonds. So this is unsaturated. Contains at least 1 double bond. So whenever you think about saturated versus unsaturated fats, saturated are usually animal fats. So things like butter and lard. What do you know about butter and lard? What are they at room temperature, solid or liquid? Solid. So they are solid at room temperature. Can someone explain to me why? Why would saturated fats be solid at room temperature?
Because they're packed tightly together due to their straight line.
Exactly. Because when these are at room temperature and there is not a lot of heat or anything from the environment, it doesn't take a lot, or it takes a lot to break these bonds between the stacks apart. So basically they are extremely stable on top of one another. So they're able to stack really tightly together. So this is multiple saturated fats. And they become very stable as a solid at room temperature because they stack really, really neatly together. Some stack so well together that even when they warm up to body temperature, they still remain pretty solid. That's not good. Saturated fats are considered like the bad fats, if that makes sense. Because if they stack together and remain solid at room temperature, they're more likely to solidify and be stored as fat in your body. Now, the unsaturated fats. So this is a particular type of unsaturated fat. Is this a cis or a trans fat? Just from looking at it, what do you think? Do it? Yeah. So we kind of talked about it the other day, briefly, about the difference between cis and trans, but we're talking about it in more detail today. So if you looked at this section right here where the double bond is, these carbon-containing groups are on the same end or the opposite end of the bond. So like, look at it like if this were straight up and down. So there's hydrogen, hydrogen, carbon, carbon. Are they on the same or opposite end of the double bond? Opposite. So this is a trans fat. So trans equals carbon groups on opposite planes at double bond. Most unsaturated fats, so if they've got a naturally occurring double bond on them, most of the time they are cis, meaning that the carbon groups are on the same end. What happens whenever you've got a carbon group on the same end? What does that do to the fatty acid tail? makes it have a little kink in it. So it makes it not lay flat. So naturally occurring saturated fats are things like oils, like fish oil or vegetable oil. Those are what at room temperature? Liquid, because they've got those kinks in the fatty acid chain and they don't stack together well. This, a trans unsaturated fat, is even worse than a regular saturated fat. Does anyone want to take a stab at why?
Because we fit more.
Because they stack on top of each other like a saturated fat, but then those double bonds even create more stability. So they are even harder to break apart once they're stacked than a regular single bond saturated fat. I'll write that down. I just wanted to make sure everybody got any pictures that they were drawn. But trans fats or trans unsaturated fats do not occur normally in nature. They are processed. They are basically that people take regular unsaturated fats like fish oils or vegetable oils, and they make double bonds. So they basically take electrons and they make them create double bonds instead of, or they make them create the double bonds at opposite ends instead of the same end. So they basically move these electrons around and change the bonding and make them to where they act like saturated fats. These are things in like processed foods and yeast and things like that, that are really, really bad for you. So they're so bad for you that the FDA requires nutrition labels to have if they have trans fats on them or not, because they're so harmful to you. They build up in your blood vessels and, cause issues. So anyway, it looks like everyone's done drawing the structure, so I'll write down those properties. So, like I said, unsaturated, so with double bonds. Occur in cis form. So that means that at the carbon-carbon double bond, the other carbon groups are at the same end instead of opposite. And so what that does to the structure is it creates a kink and makes it not stack as easy. I'm going to put good. And then some examples. So vegetable oils and fish oils. Trans unsaturated fats are synthesized, made by man, and are so bad So it talked about there are actually some countries that have limited or even outlawed the use or synthesis of trans fats. Do you want to take a stab at why people would do that? Why do you think people would mess with unsaturated fats and make them worse for you? What do you think it does? Does fish oil taste very well? Or good, I guess? It doesn't. So basically, whenever they change this chemical reaction, it can make it bind to taste receptors differently and actually taste better. And so they tried to make it taste better. America. Okay. And again, these are found in baked foods and processed foods, things like that. So it can also help keep something better for longer than naturally occurring fats. So it wasn't all just about taste. It kind of make it last longer. Okay, so that's all on fats. What do you remember about phospholipids?
It only has 2 fatty acids.
It only had 2 fatty acid chains instead of 3 like a normal fat. So they're called triglycerides because they've got 3 fatty acid chains. Phospholipids, a couple different things. So they only have 2 fatty acids. And then what's special about the glycerol backbone? What does it have tacked onto the end of it? Not quite. I'll give you a hint. It has to do with the phospho name.
Phosphorus.
It's got a phosphate, which contains phosphorus. So you were like 25% correct. So what's special? So as I said earlier, we've got glycerol and then 3 fatty acid chains. And this is our normal fat. A phospholipid has your glycerol, and then at the end, there is a phosphate. Mind you, this creates a negative charge, and then only 2 chains. So this is a phospholipid. So that's special because it does a couple different things. We all know fats don't dissolve in water because both the glycerol and the fatty acid chain don't have like polarity to them. They're a very pretty stable and uncharged molecule. Whereas the phosphate, I drew that little negative here because a phosphate has 2 oxygens that are readily available to bind with other things, making it a polar molecule. It's got a polar end and a non-polar end. So how do you think it acts in water?
One like water.
One likes water and one doesn't. Which end will like water? Polar or non-polar? If fats are non-polar and don't like water, the polar end is the end that likes water. So, and I'll put this over here. So I'm going to draw a phosphate. So that's what a phosphate is. And you've got your glycerol. I don't care that you know the chemical components of a glycerol. That was the little 3-carbon sugar that we drew over here. That's just glycerol. So I'm not going to draw that over and over again. And then your fatty acid tails. Now, the tail on one end is also kinked in cis form, so that even makes it better at staying stable and not stacking on top of one another. Or I say stable, staying stable separately from one another. It's not like these phospholipids are going to combine with one another and stay stuck together. It's very fluid. Do you remember the term fluid mosaic model when we talked about the cell membrane? It's been a couple years, so I understand if not. But the fluidity of a cell membrane has to do with this kink in that bond right there that makes it to where these fatty acid chains on a lipid don't stack on top of each other. So this end right here is polar, meaning that it is hydrophilic, so water loving. This fatty acid part is nonpolar, and it is hydrophobic. So I already kind of cheated and gave you the answer, but what does it do when in aqueous solution? Meaning that what does it do if you put a bunch of phospholipids in water? What are they going to do?
They assemble into bilayer.
Into a bilayer. So that is super duper important. I'll make sure everybody gets this information before I turn it over and draw a bilayer. But a lipid bilayer, a phospholipid bilayer, is what makes up all of our cell membranes. And the reason that it does that is because this nonpolar hydrophobic end wants to be away from water, and no matter where you turn it, if there's still water around it, it's going to want to repel it. So all of these phospholipids together will create a bilayer to protect the nonpolar hydrophobic ends from water, and then these will bind with the water. So, oh, sorry. Just holler at me if I'm going too fast. But not too loud. I'm sensitive.
I don't think you're going too fast. I'm just.
That's okay.
Speed up camera. Yeah.
All right.
No.
Oh my God.
Okay, we're good.
Okay. So if you've got water, so these are our hydrophilic they're referred to as heads. That's where the phosphate is at. And then our little fatty acid chains here all hang out in the middle. Has anyone ever heard of micellar water? I don't know how to say it. Okay. So the reason that it's called that is because it contains phospholipids, or it contains lipid. I don't know if they're specifically phospholipids, but it contains lipids that have both a hydrophobic and hydrophilic layer. So what they do is they form what's called a micelle. So it's a little double layered bubble, if you will. And they can actually like pull the dirt and grime, and the chemical reaction that's going on is it pulls that dirt and grime into the bubble, and then that bubble washes away whenever you wash it away with water. Super neat. So there's chemistry behind your fancy dancy face care. It's kind of like magic water. But if you pour micellar water into like regular water, then you'll see it bubbles up and it separates. That's because it has lipids in it. So,
can you drink?
I mean, I wouldn't.
What happens?
I don't think anything would happen.
Mango.
It's like
Because imagine all of the phospholipids we consume. Every single cell membrane has phospholipids. Our body would just get rid of them. So this right here is a phospholipid bilayer. And this is all cell membranes. Even plants have cell membranes. They just also have a cell wall outside of them that contains things like cellulose and stuff that we can't break down. It just adds more structure. But we encounter lots of cell membranes in all of our foods because every cell has them. So, another important note, as I mentioned earlier, the tails have a cis bond kink in them. Making the membrane fluid. So it doesn't stack together. And that's super important because, as you know, all of our cells have cell membranes, and what has to happen between cells? Does all the stuff in your cells just stay in there?
Yes.
That answer's no. So cells have to, think of them like factories. There's stuff that comes in, there's stuff that goes out. All of these cells are switching materials and letting ions, so nerve cells use ions as a signal molecule to tell it what to do. Those ions have to move across membranes. Your ATP molecules that have to be broken down have to move in between these membranes. So there's all types of stuff that is moving in and out of membranes. And so they need to be fluid. Does that make sense? Yes, no, maybe so?
Yeah.
Okay, awesome. Okay, the last group is steroids. There's not much that we're going to talk about with steroids, just basically why they're considered lipids, because some people really don't know how to classify steroids, because they kind of don't look like a normal fatty acid, or not a fatty acid. They don't look like a normal lipid, but then they're not like nitrogen containing or anything like that, like a protein. So they're just a little weird. But with steroids, the main thing that you need to know is it has 4 fused rings. And these are just carbon, hydrogen, and oxygen. So again, they don't contain nitrogen or anything like that in these rings that make them act like proteins. And then they have a single short fatty acid. So the picture, again, I'm not going to draw it because it's okay if you don't even have the picture in your notes. It's basically just a couple of hexagons and a pentagon, or a few hexagons and a pentagon, and then a very short little hydrocarbon tail up at the top. These are, again, considered lipids because they technically have a fatty acid and they technically have a carbon, hydrogen, oxygen backbone. It just looks a lot different. These are usually used for signaling, so they're signaling molecules. Cholesterol is one of the main ones that you've probably heard. You've probably heard of cholesterol as being bad. Well, there's good cholesterol, because you need it in your body, because that is what we use as basically the basis of things like testosterone and things like estrogen, and other steroid type signaling molecules. And again, that's all that they are, is they're just signaling molecules. There's receptors in certain cells that accept them. Like for instance, estrogen, there are receptor molecules in a female reproductive system that whenever there's so much estrogen or too little estrogen, it will affect like ovulation cycles and things like that. All of that has to do with signaling, and that signal molecule is estrogen, which is a derivative of cholesterol. So anyways, cholesterol isn't all bad. Does that make sense? Okay, so that's pretty much it on steroids, is just basically why it's considered a lipid, because it's not technically able to be classified as anything else. Okay, so where are we at with time? 12:36. So what we will do now is, if you will log on to your AP College Board. Let me make sure. I might not have it posted yet. Hold on. Let me change. I might have set it for 12:45. Stop recording.